Morning, everybody. So my role here is really to make a general introduction. As chair of the Industrial Advisory Board, my role is to really encourage the local industry leaders to participate in that board and provide a connection between industry and the academic groups here in bioengineering. Our group extends up into Orange County and, of course, the San Diego area. We like to see a connection between industry and the faculty, industry and the students. Bear in mind that we're the employers of all of you, and so we really want to encourage you guys to be educated in a way that really works for us. So we're happy to give back. As part of that, we are sponsoring this part of this session, and so I get the envious role of introducing the keynote speaker. I want to congratulate you all on the organization of today's meeting. This has grown over the years, and today I understand we have a record attendance of over 450 registered students, which is immense, and you really deserve great congratulations for organizing that. Phenomenal achievement. The second point of congratulation, over the years we've also encouraged you as students to try and recruit speakers that reach outside the closed environment of a university setting. Today, you've recruited as your keynote speaker Craig Venter, and that is an amazing achievement, so congratulations on that. You're going to hear a story which will really open your eyes. Now, my job is to give you a brief introduction of his background, so you can put it into a context. So, of course, he had obtained his bachelor's and PhD from no less a place than UCSD, and then went off to be a professor at SUNY Buffalo. So he's paid his dues, because it's a long way away, and it snows a lot up there. I've been, but I'm not going back. He then went to NIH and became section chief, and showed his skills scientifically because he invented ESTs, which is a massive driver in developing an understanding of expressed genes. He left there to become founder of the Institute for Genomic Research, or TIGR, and they did some impressive work and decoded the first genome of a free-living organism. So that was an immense step there. Then he became founder of Celera Therapeutics. The objective of this, as I understand it anyway, was to sequence genomes, particularly for the human genome. Now, this was a big step. What Craig did was not only be an inventor and an outstanding scientist, he was something of a maverick here, because he recognized that the way we were going about sequencing the human genome was in a stepwise linear way that good scientists, good engineers usually do. And if we'd gone down that pathway, we'd probably still be doing that sequencing today. He set out to do it a different way. And when he said he was going to do that in the time frame he was going to do it, it caused an immense stir and created a race. That race resulted in the genome being sequenced much, much more quickly. And while it turned out that it was a race, they agreed in the end between the NIH-type organization and Craig, they published together. So everybody won. That achievement was absolutely immense. So he's gone on then, and he's now founded a new institute, and he's also a founder of synthetic genomics. He's, along the way, picked up a large number of academic and scientific awards. There's some of the highlights here, which includes the U.S. National Medal of Science. What's interesting is not only has he picked up the scientific side of the awards, but he has a personality that reaches out into the general public. And most of us as scientists, we live in our own little bubble, and that's where we stay. his personality reaches out. And so he's had some additional awards. And here's a few that you might be interested in. 2010 Vanity Fair's 100 Most Influential People of the Information Age. Time Magazine Top 100 World's Most Influential People of Scientists and Thinkers. America's Best Leaders 2005 US News and World Report. And a couple that I really like down here. Men of the Decade in Men's Health. And Man of the Year 2005, I think, by GQ. Ladies and gentlemen, I have to present Craig Ventner. He's going to tell us about digital biology.
Well, thank you for the kind and unusual introduction, and I'd like to thank the students for inviting me here. I actually decided this was a good thing to do on a Saturday morning, even though I have a lot of other things to do, but this is a good place to start, at least till the sun comes out. So I'm going to, I have to get this started down here, tell you about digital biology and genomics. But first, let me introduce my institute. So the top left is our main campus in Rockville, Maryland. And this is where we molded the Institute for Genomic Research and several other not-for-profits all into what the trustees now call the Ventner Institute. And we also have a campus on Research Drive in La Jolla. And you can't have missed our new building going up on the corner of Torrey Pines and North Torrey Pines. And those of you, the engineers in the traditional sense, will like this building because it incorporates lots of new engineering design principles. It's going to be the first carbon neutral research building in the world. It's going to generate all its own power. We're going to recycle all the wastewater, but we couldn't get a permit from San Diego to feed that water to students as a test, but we might get that in the future. So it's a living experiment, and it was very tough to get all the things to have an active research lab with fume hoods and massive banks of computers, so we're water cooling the computers. Simple things as we put the fume hoods in small rooms so we don't have to turn over all the air in the entire building just because you have a fume hood and some OSHA requirements. So lots of unique principles going into it, but I think it also has a great view of La Jolla Cove. So we hope a number of you will end up working down there when it's after it's finished this fall. So I'm going to talk about the first stage of this, and that's what I call digitizing biology. And that's where we convert the analog code of DNA into the digital code in the computer. And this is pretty straightforward and has been going on for a long time in a variety of ways, but these databases are growing substantially, as you heard in the kind introduction, when we sequenced the first genome in 1995, and then 5 years later, scaling up to the first draft of the human genome. Then 2007, the first diploid genome, which happened to be my genome, but so while my genome's the human reference, I'm not sure I'm the correct human reference. But depends on what your goal is. And now this technology is changing dramatically. So we now have at the Institute high throughput single cell sequencing where we can do a genome on a single bacteria or a single human cell, a stem cell. And we've been using this, for example, to do haplotype separation of the human genome. Now, everybody knows that you got a set of chromosomes from your mother and from your father, but there's nothing in the genomics community that takes advantage and measures that information because they haven't been able to. And so one way is to separate the digital information back into the haplotypes of your parental chromosomes is to sequence sperm cells because they're a haplotype genome. And it turns out there's an average of one crossover per chromosome per sperm. And no two sperm cells, no two egg cells are alike. They all have crossovers in different points, and people are always amazed when their children come out so different. Genetically, each combination is a totally unique, untried experiment. But by isolating and sequencing single sperm cells and doing just a few of them, you can easily separate the genome into haplotypes. Now, if you don't have sperm, it's a little bit more complicated, but not so much so because somebody in your family did have sperm, and so it's easy to work back. And once you have a haplotype phase genome in your family, you can sort out the rest. And the significance of this is instead of the simplistic view that's often presented about genetics, that everything is down to just a single change in a gene, and if you have that change, you either have a disease or a trait or you don't. It turns out basically there is no standard gene or protein sequence. It's not all the same in all of us. Basically, our whole repertoire is a little bit different in everybody. And in almost every gene and protein that you have, the genetic information you inherited from your mother is different from what you got from your father. And so these are called compound heterozygotes because you can have two different variants. And so for a typical protein, you can have at least two different versions of it, and often more depending on splicing and other things. And it turns out these compound heterozygotes are more often associated with complex traits than the simplistic view that's been out there. That's almost all I'm going to say about genetics per se, other than our gene repertoire is the small part of what we have in our bodies. We use the same shotgun sequencing that we use for sequencing the first genome and then the human genome to start this new field called the microbiome. And Karen Nelson at the Institute just did a shotgun sequencing of microbes from the intestine. Now this is expanded into a huge field looking at microbes in every different part of the human body. And so in addition to our 22,000 or so human genes, we have about 10 million additional microbial genes. And so if you just think about it, about 1,000 different microbes and millions of different genes just in your mouth. Think about that the next time you're coughing or kissing somebody. All the genetic information you're exchanging. And so the challenge has been to understand the role this plays in biology. And it turns out it's now becoming hard to find a disease and a set of human traits that's not affected by your microbial metabolism. And this is a field that's expanding very rapidly and gives me optimism for the future because we're starting to get all the components now of human biology to be able to understand things versus everything being treated like a black box. For example, just trying to understand what's the metabolic potential of these 10 million or so genes and what impact does that have on your physiology. And just a simple set of calculations and some now real-life experiments show that our protein repertoire can manufacture about 2,400 different chemical substances. So it's starting to get quantitative in that sense. So we can simply ask the question is, after you have a meal, how many different chemical substances are circulating through your brain? And it turns out there's on the order average of roughly 500 chemicals circulating in your bloodstream right now. But only 60% of those are from human metabolism. 30% are from the different chemicals from your various species you consumed in your diet. But 10% are on the order of 50 chemicals right now circulating through your brain are from bacterial metabolites of those chemicals from your diet and from the human chemicals. So nobody has any idea right now whether those affect how you think, affect your disease state, improve exercise tolerance, et cetera, except now it's starting to come up where these bacterial metabolites very much affect possibly whether red meat can produce damage to your heart through a bacterial metabolite. A nice study that Karen Nelson did with David Brunner here at the medical school is showing that alcohol liver damage is not actually caused by a direct effect of alcohol in the liver. This gives me great comfort. In fact, it's due to effect of alcohol on a certain bacteria that releases a toxin that damages the liver. So I've been arguing for some time that we should fortify alcohol with antibiotics, but that has not caught on yet as a major medical practice. But it starts to show that things that we've assumed commonly in biology and medicine are due to something very different than we had thought prior. And turns out bacterial toxins are some of the leading causes of certain types of cancer. So, and I'll get to this later, targeting specific microbes. But understanding the microbiome is key to understanding biology and medicine. But one of the biggest challenges is it gets cheaper and faster to sequence human genomes and microbial genomes, is that data in the absence of phenotypic information is basically useless. So I think the biggest challenge for the entire scientific community right now is trying to define a human phenotype. Just think if you had to write down all the information associated with your life, your chemical composition, all the x-rays and other tomographies that you've had, all your physiology, how your thought process works different than your family members, people sitting next to you, et cetera. And there is no standard for this. People have tried to do this in some specialties of trying to define cardiac muscle phenotypes. People working in the eye have tried to define phenotypes around that. But it's virtually impossible right now to get together a community view of what a human phenotype is. Until we progress to what, that sounds very simple to do, genomics in medicine is going to be limited in how it progresses. We've used these same tools to do a shotgun sequence in the ocean, and this data has greatly expanded the gene sets that we all have to work with. People consider this quite often my best invention because I found a way to sail my own boat around the world and do science at the same time to justify the trip. People in the Department of Energy in particular that work in acid mines and things were envious of this approach. But this is sort of the various routes that we've taken over the last several years, covering over 80,000 miles at sea on a sailboat and taking samples every 200 miles. It's totally changed the view of the microbial world that was out there. When we started this, ocean microbiologist numbered the organisms starting with 1, and they didn't get much further than 20. And people were sure there was very little diversity in the oceans. In fact, SAR-11, the 11th organism discovered in the Sargasso Sea, even though it was thought to be abundant globally, was thought to be a single organism. And what we've done with shotgun sequencing, each of those little bars represents about 900 base pairs of DNA sequence. And what's across the top is a reference genome, and it almost doesn't matter which one, because you can see instead of just a few % variation that you might expect, all humans, we all differ from each other by 1 to 3% in our genetic code. All mammals are a maximum of 10% different in the genetic code. Here we have variations that will look like a single, a subset of organisms going up to 50% sequence variation. But because we have 900 base pairs of sequence, and a typical bacterial gene is 900 base pairs, and these are random fragments, we get on average 1.4 genes per sequence. And so we can actually get gene order going down the chromosome. And in all these variants, gene order is largely identical. You can see some gaps there that large gap on the right is due to the reference genome having an inserted viral sequence, so it doesn't show up much in other sequences. But it's tremendous diversity. And just to make a very long story short, as we accumulate more and more data, we found that what people thought were single organisms are entire taxa. And in fact, new taxa that people didn't even know existed before. And some of these first view of these organisms came from compiling the genome sequence from these millions and millions of sequence reads. So it's a very different view of what's in the oceans, but we've done the same things with the air. During the course of this lecture, you're going to breathe in and out about 1 million microbes and up to 10 times as many of that viruses. If you swallow a mouth of seawater, each milliliter has about 1 million microbes and 10 million viruses. So we are part of a microbial universe. Our bodies are inhabited by a substantial number, and I think it's important to understand the life of the planet to understand these microbes. So all this type of sequencing has filled the databases with digital information. In fact, it was easy to get an NIH grant to generate more digital genetic information. It's been very hard to get them to understand that digital information. So I think that one of the most important fields going forward will just be the computational analysis of this data and what it's telling us. But we've tried to take this in a new direction of a way to understand the information by starting with the digital code, regenerating what I consider an analog molecule, DNA, and seeing if we can recapitulate biology and life from the computer. We started this early on after we sequenced the first 2 genomes and comparing them and just asking simple questions of how many genes are essential for life? Can we come up with a definition of a minimal gene set that defines life? And ultimately, we decided after trying all kinds of experiments, the only way we thought we could get there was by trying to design and construct a chromosome with the gene set in it that we wanted. So that was sort of the origins of this new field of synthetic genomics. And it took us quite a while to get there, in part because as soon as we decided down this route, there were new questions. Would technology even allow us, would the chemistry allow us to even make really large DNA molecules the size of chromosomes? DNA is very brittle. As it gets larger, the DNA synthesizers create lots of errors. And then the second question was more of a biological one. Even if we could make this large chromosome, could we find a way to boot it up in a cell? So we started down routes with both processes. We decided to start with something small to see if we could correct the errors in DNA synthesis. So we decided to start with something historic, and that was Phi-X174. That was the first DNA virus genome. It was sequenced by Sanger's group. It wasn't the first viral genome. The first one was an RNA virus genome the year before. But this in part contributed to Sanger getting his second Nobel Prize, because this was the use of what's now called Sanger sequencing to do this genome. So we decided to start back with this genome because it's known that Phi-X can't tolerate many errors and still get a virus out of it. So we synthesized the 5 kb piece and used these new error correction tools that we had at the time and injected that DNA into E. coli. And the next stage was exciting, and this was the first actual plate, is obviously E. coli read this piece of synthetic DNA, made the viral proteins. The viral proteins self-assembled, made the viral particle, and the viral particle showed its gratitude by killing the cells that generated it, and that's how we detect it. This is a lawn of E. coli, and every place you see those clear spots is where the virus killed the bacteria. So we call this a situation where the software is building its own hardware. All we put in the cell was a piece of synthetic DNA hardware, and we got out viral particles that can infect other cells, reproducing themselves and killing the cells when they lyse it. So we're very encouraged by this, but we didn't want to make small viruses. We wanted to make an entire bacterial chromosome to see if we could recapitulate life. But we thought now that if we could make pieces the size of viral genomes, we could find a way to put them together. So we started down this road, and this was it took many years to get there, but finally we settled on a process of synthesizing viral-sized cassettes of around 5 to 7 kb, and then we would put 4 of those together to get these 24 kb pieces. And at each stage, we would clone these into E. coli, produce a lot, resequence it, make sure that we weren't inducing errors as a part of the process. And if there were any errors in the sequence, we went back and corrected them. The next stage, we put 3 of those together to get these 72 kb pieces. And at that stage, we more than doubled the world record for the largest piece of DNA that had been synthesized, which was around 30 kb. And then the next stage was putting 2 of those together to get quarter genome-sized pieces at 144 kb. And at this stage, we ran into problems with E. coli not liking these large pieces of synthetic DNA. So we looked around for another species to clone them in, and we found that yeast, Saccharomyces cerevisiae, makes bread and beer, so it's one of our favorite organisms in the history of humanity, readily takes up DNA pieces and can clone them, and even better, puts them together by homologous recombination. So simply by putting the 4 quarter synthetic pieces with a proper overlap and a small vector, and the vector just has an origin of replication and a yeast synthetic centromere. And putting these pieces individually in yeast, it assembles them together automatically using homologous recombination. So this resulted in our first complete synthesis of a bacterial chromosome. This was around 600,000 base pairs. This was a Mycoplasma genitalium genome. And at the time we published this about 5 years ago, this was the largest chemical of a defined structure ever made. And obviously we sequenced it and validated it. But the next stage was to see if we could boot that up. At the same time, we had people continuing to work on improving the synthesis. One was the biggest advancement came with at the time, a young postdoc named Dan Gibson, who's now a vice president of synthetic genomics, taking this to a whole nother level. And what he found is everything we were doing as individual steps could be combined into a single tube at a single temperature. So this is now called the Gibson assembly. People are buying kits from New England Biolabs and other places, and you just simply put in the oligonucleotides, if they're designed properly with the right overlap, put in this tube, and you get out the right pieces in the end. And what this means is this can now be automated. So we can actually go from the digital world to making these analog molecules in a completely automated fashion. And I'll show you some results of this in a while. We had a second team start to work on genome transplantation. And it turned out this was a harder problem to solve than the synthesis. Just nobody had tried to tackle the synthesis before, so we thought it was going to be the hardest problem, and that's also why we chose the smallest genome. But doing transplantation was a lot more difficult to work out. But this paper that we published in Science in 2007, I think is one of the most important ones that we've ever published that leads to a basic understanding of biology and how life works. And because of its importance and importance to the future of synthetic genomics, I'll walk you through what we did in this study. So we chose 2 Mycoplasma species, and we chose Mycoplasmas because they don't have a cell wall, so it's easy to get DNA in and out. And we chose two species that were roughly the same distance apart that we are from mice, a little over 10% in this case. And we isolated the genome from M. mycoides. And when we were starting this work, we had no idea whether there would be proteins required for transplantation. Do you have to have single-stranded binding proteins? Do you have to have, the equivalent of histones with eukaryotic genomes for folding the DNA, or could we use just naked DNA? So part of the early experiments involving learning how to do transplantations was we treated these genomes with proteinases to remove all the proteins, but we had to develop other techniques because you can't pipette these genomes. Large pieces of DNA are so fragile, just pipetting them causes shearing of the DNA. So we isolate everything in small gel blocks. We can infuse enzymes in and out to deal with the DNA, to deal with the proteins, and deal with them analytically. And we added a couple cassettes, one so we could select for whatever cells had this chromosome in with an antibiotic selection, and another just so in the presence of the right chemicals, with the cells would be bright blue. And we developed ways to insert the genome into the recipient cells, M. capricolum cells, which have a smaller genome with enough differences to make it very clear cut. So we have this movie to show you what we think happened. We inserted the M. mycoides chromosome into the capricolum cell. Now we have an unusual situation of a single species with two sets of genetic software. And one of the things that we think happened, as soon as we put in that DNA, it immediately started to be read by the enzyme systems and started producing proteins. Some of the early proteins produced were restriction enzymes that my colleague in all this work, Ham Smith, got the Nobel Prize for discovering in 1978. And those restriction enzymes recognize the chromosome in the cell as foreign DNA and chewed it up. So now we have the body of one species and the genetic software of another. And what happened in a short while, we ended up with these bright blue cells. And when we interrogated them, first with 2D gels and sequencing the proteins, there wasn't a single trace of anything from the capricolum cell left. All the proteins now in this new cell were what was coded for by the M. mycoides chromosome that we transplanted. When we tested with antibodies, checked every aspect of the cell, there was not a single molecule, as far as we could tell, from the capricolum species. So that's why life is a DNA software system, and when you change that software, you change the species. This is a very important concept for understanding the fundamentals of how biology actually works. And believe it or not, this was a major surprise to a not insignificant portion of the scientific community in molecular biology who thought that these experiments would not be possible. Well, I could take you through lots of technology of building to the next step, but using the Gibson assembly, using some new, very advanced error correction that gives us correct molecules without sequencing, we decided to change tack and build the entire M. mycoides genome. There's lots of steps in between. We had to learn how to isolate the genome from yeast to do the transplantation. It actually set us back 2.5 years because it turned out this genome in the mycoides cell is methylated, and the eukaryotic cell does not have the methylation enzymes to methylate it. So every time we did the transplantation out of yeast, the genome was just chewed up by the restriction enzymes in the cell. So we isolated all the methylases, we methylated the DNA, and those chromosomes could be transplanted. And ultimately, we removed the restriction enzyme genes from the capricolum genome, and then we could just transplant naked DNA out of yeast. So the restriction system is very key to the microbial world, and you can understand why. If you just take up your neighbor's DNA, you become your neighbor. So if you don't want to do that, you have to have some degree of protection. That protection is the restriction system. just imagine if you're petting your pet dog and you absorb some DNA and your cells became converted into canine cells. That's what happens in the microbial world, and it happens all the time. So we synthesize this starting with 1 kb pieces. We put 10 of those together to make 10 kb pieces. We put 10 of those together to make 100 kb pieces. And we put 11 of those into yeast that assembled the entire chromosome. We did the transplantation, and it didn't work. So as software engineers have debugging software, we had to create the biological version of debugging software to find out why this chromosome didn't work. And basically, we tested each 100 kb piece one at a time, and we found one that didn't support life. We went back and resequenced it with Sanger sequencing. We had used next-gen technology, which can be very consistent, but just fundamentally wrong. Each new technology has its own biases. And there was a single base pair deletion, the DNA A gene, an essential gene for unwinding DNA. That single base pair deletion made the difference between life and no life. We corrected that one letter out of 1.1 million, did the transplantations again, and got the first synthetic cells. So this was in early 2010, and obviously there was a lot of news about it, and the paper was published in Science. And what we disclosed in the Science paper is that we put watermarks in the DNA to prove that it was totally synthetic, but we said that people had to solve the puzzle of what the watermark said. These are the watermarks that we put in. I don't know if the color is a little bit off here, but we invented a new code for writing the entire English language with numbers and punctuation using the 4-letter DNA code. And if you solved the first watermark, it gave you the code for solving everything else. And being the first genome designed in a computer, it had a URL built into its genetic code. So if you solved this, you got the URL, and you were instructed to send an email to the species saying that you solved what was there. And after a certain number of people did that, we posted the complete thing on the internet. So the watermarks contain not only the new code, and it's very important. A lot of people, there's been a lot of press on this lately of people writing things in DNA, and people have been doing this for a very long time, just using the ASCII system. But it's not biologically supportive. Our code that we develop puts in very frequent stop codons, because the last thing you want to do is put in a name or a quote from the literature and have it create a new toxin that kills the cell or others. So our code puts in very frequent stop codons so we don't get any peptides. So in addition to the names of the institutions and the 46 scientists that contributed to this over time, we put 3 quotations from the literature that I found that I liked. So the first one, to live, to err, to fall, to triumph, to recreate life out of life, from James Joyce, seemed totally appropriate. The second one is from American Prometheus, Oppenheimer's biography, see things not as they are, but as they might be. And the third from Richard Feynman, what I cannot build, I cannot understand. Once all this information became public, one of the first responses we got was a letter from James Joyce's state attorney asking us if we had copyright permission to use his quotation. Under American law, you can use up to a paragraph without permission, so we dismissed that one. And then we started getting emails from a Caltech scientist who said we had misquoted Richard Feynman. He unfortunately had died, so there was no way to check it with him. And if you look on the internet, this is the main quotation you find. But to prove his point, the scientist sent us a picture of Feynman's blackboard with the original quotation that I guess one of his biographers messed up. So the original was, what I cannot create, I do not understand. We think it's a much more appropriate quotation. So we've gone back to change the genetic code so that Feynman can rest peacefully. So we've proven what had never been proven before, that DNA in fact contains all the necessary and sufficient information for life, and that you can now write that code starting with the digital information in the computer. Since then, we've been working diligently to shrink the genome further and further, and we've been talking to Shu Chen and others about creating a course here on genome design. That the challenge is, even with our minimal gene set, which is almost half the size of the original genome, about 50 of the genes are of unknown function, other than that they're essential for life. So the problem is we don't know enough about biology to do what we'd like to do, just sit down on the computer and design new life forms. All we know is that those 50 genes, if you take them out, you can't get a living cell. So biology, in our limited knowledge of biology, is a major constraint in the future of design. So empiricism is going to be a big part of this. But we can start to re-look at life in a different way. And we like to use the computer analogy. So one of the major experiments that we're doing right now is we're trying to defragmentize the genome. After 4 billion years of evolution, genomes are kind of messy things. And so we're trying to see if we can group genes in cassettes by function to make design easier for the next stage. And so we're trying to build some of these right now, so we'll find out if that's possible. The trouble is the design is when you go from the origin of replication, you have to have a certain orientation of genes. So it's not a matter of just lining up the right genes and the right cassettes. You have to have them on the right strand of DNA. So a lot goes into design. So a design course would not be trivial, but we were going to propose that whoever designed a genome, we would build it, and if it created a life form, you'd get an A. I don't know what happens if you always fail, but so we're just at the first stages now of totally dealing with digital biology, dealing with digital design, and I'll just show you some quick examples of some directions we're going. And we have about 80 million genes now in the public databases. The majority actually came from the deck of our sailboat, from shotgun sequencing the ocean and the air. And we view these as design components. The same way the electronics industry in the 40s and 50s had resistors and capacitors, then transistors, then integrated circuits. Everything we know about the digital world is with a couple handfuls of components. We have 80 million right now, and it's growing. We don't know where it's going to tap out, 400 or 500 million unique genes to work with. We have software at Synthetic Genomics for actually trying to design new cells based on what we want in them, but it's going to depend on how well this modularization and defragging works. We're trying to design a series of cells to use sunlight as their energy source and CO2 is their main carbon source. And the whole field now that we're trying to drive is this shift from digitizing biology to going the other way, starting with that genetic code. We just recently formed a new group at Synthetic Genomics, SGI DNA, because it's the only place in the world right now that can build these really large constructs. And we're doing that for a large number of groups now to really advance what people are trying. And it's like the early days of sequencing. After we sequenced the human genome, everybody was sure there was no longer any need for DNA sequencers. It's really comical when you look back at it, but everybody starts to think when a threshold is passed that that's the end, but to me it's the beginning. That was the beginning of the phase of sequencing. This is the phase of genome design, and as people have the capability, it's changing. For example, we can build multiple components, like thousands of variants of a gene or a protein, and test them in a combinatorial fashion. We're trying to build a computer operated system that is self-learning, that can actually go back and make changes and learn 10,000 times faster than a graduate student. Just think how much you could accomplish if you had some of these tools now to examine what we don't have enough scientists on the planet for, is functionating soon to be hundreds of millions of genes. We have to have new approaches for doing that. We're using these to understand the genetic changes in stem cells. So Larry Goldstein's lab made some iPS cells from a skin biopsy from my arm and made some neurons that some of you may be working on. So he said my cells are taking over his lab and other labs. I told him I feel my consciousness constantly expanding. But as soon as you put cells in culture, the genetic code starts to change. And obviously that's not a good thing if you want to turn around and use these cells as replacement for human therapy. So we've been working on ways using synthetic DNA approaches to go back and rewrite the genetic code in stem cells to create new functions. So DNA synthesis, DNA design will be a future part of human therapeutics in lots of unique fashions. In terms of industrial applications, when we look at what's potential with algae cells, they greatly exceed anything in the existing plant world in terms of productivity. And with getting some good changes genetically, the potential is tremendous. At Synthetic Genomics, we have a program where we're making a synthetic algae chromosome to add totally new metabolic functions to the cells that they've never had before. And by the way, this is how cells acquired most of their functions. Our mitochondria were from a bacteria that got absorbed into eukaryotic cells. The chloroplast was an early photosynthetic bacteria. We've had some success with engineering the photosynthesis pathway, improving the efficiency of light-activated processes by just reducing the antenna size and changing the shading in cells. But I want to, with the end here, talk about what I consider sort of the proof of the digital conversion of biology, and it's something we're calling biological teleportation. And this is not the beam me up Scotty version. While quantum teleportation is starting to become real, it's way too complicated in terms of moving humans. But we can send digital information and convert it back into biological information. So we could, for example, get a biological sample from Mars in as little as 4.3 minutes when Earth and Mars are at their closest. This is in contrast to try and get a rocket up there that's big enough to take samples that could blast off from Mars and get back to Earth. You may not be aware of it, but there's all kinds of concerns and rules about bringing extraterrestrials back to Earth. And there's whole societies that have great concern about this. And you might remember or have seen, because probably a lot of you are too young, but with the first people returning from Mars, they were put in supposedly bacterial secure mobile homes to make sure they didn't bring some strange contaminant back from the moon. So if we're just doing digital information, we could actually reconstruct the alien life in the laboratory. We're using these processes to come up with new ways to deal with antibiotic resistance. We're actually making synthetic phage to target specific microbes. So a phage, we can easily send that digital information through the internet and just have a box to convert that back into making the DNA that could infect and kill a targeted bacteria. With vaccines, the first genomic vaccine just came on the market in Europe. It took 17 years from when we started the process. This is against type B meningitis. So we were looking for ways to speed up the process, and we decided to work on the flu virus and approaching it with synthetic DNA. Just to remind people, 1918 flu killed about 3% of the Earth's population Today, that would be almost the entire population of the US. And currently, to make the flu vaccine each year, about 900 million chicken eggs are needed. So it's not quite going back to Pasteur technology, but close to it. My institute's been part of a sequencing surveillance network, and we just recently published the 10,000th flu virus. But the process we've been developing is to take that digital information and immediately make a vaccine out of it in a very short period of time. And we've been working with Novartis and with BARDA in the US government. And what BARDA sends us is an email of a test pandemic sequence, and our teams have less than 12 hours to make that new vaccine and get it to Novartis. And so we've developed this digital biological converter to do this very rapidly. And we got a real world, whoa, I lost it. Where did everything go? PowerPoint's encountered a problem, needs to close. That's an interesting way to have people finish their talk. With this new virus that's just recently emerged in China, the CDC couldn't get a hold of the virus, but the Chinese did something that was surprising and very positive. They actually published the sequence of the virus very quickly because they were worried about getting help. We downloaded the sequence, made the new flu species, got it to CDC and Novartis, and a vaccine's already in development. You might remember with H1N1 that you all got shots for, there wasn't a vaccine available until 2 months after the peak of the pandemic. I just have a couple slides at the end, so let's go to that one. So this is just the recent thing we did with the new H7N9 virus that's now, as of this morning, has killed 44 people in China. So there's concern about it spreading rapidly. We got the first sequence on March 31st. On April 1st, we had the first gene synthesized, and by April 6th, the complete virus was available for the CDC and Novartis. So we've actually built this machine that's a complete digital biological converter that can just take digital information and spit out. We can make proteins, viruses, and some living cells. And we're going to be testing this later this year on a C-130, see if we can send a sequence up to kill a bacteria on a C-130, showing that it is just digital information. We're putting one of these at Novartis's new cell facility in North Carolina. This is where they're using MDCK cells that were actually, boy, it's raining water here, developed by Gordon Sato's lab in the 70s here. And they're using this now to produce the vaccine. So with just having a digital biological converter, all we need to do is download a sequence at that site, and it can go right into vaccine production. But why not have lots of these? The Novartis facility can make 300 million doses. That's not enough for 7 billion people. Even when the vaccine for H1N1 came out, there was only a priority of health workers, senior government officials, et cetera, who got that vaccine first. We can send that information around the world in less than a second. If there's multiple centers, if UCSD had its own digital biological converter, it could make vaccine on the site. And the future is having a box attached to your computer where you can download insulin from the internet, you can download a vaccine, you can download a phage to target specific microbes. Biological teleportation is going to be a key part of the future of medicine and how we deal with things. Biology used to be controlled by whether somebody would send you a clone. Now, with DNA synthesis, most people don't bother to even get something or clone it. You download the digital information, you synthesize it. That's going to change from just the gene level to, in the future, synthetic stem cells, et cetera. It's a pleasure to be here. Hopefully this will excite some of you to take on these new ideas for the future. Thank you very much.
Dr. Ventnor will take a few questions if you have any. If you don't have any, you weren't listening.
Actually, I don't think we unfortunately have time for questions. But I'd like everyone to join me in thanking Dr. Ventnor once again for giving such an inspiring speech. And depends on whether we want to cut into lunchtime.
You always hate to get between people and food, but if you're not serving alcohol, it's probably easier.
If anybody has a pressing question.
Thanks. That was a fantastic presentation, very kind of mind-blowing. So I am curious, you've probably thought about this before, but I guess with all the great uses of all these technologies, there could also be the potential for other countries perhaps doing bad things with them. And I wonder if you have any thoughts about that.
No, it's been a very key part of things from the beginning, and I left those slides out, but we started asking for ethical review before we even started the first process, and I think it's the first area of science where there was lots of discussion, reports published before the science succeeded. And the most recent one was when we announced the first synthetic cell. President Obama's Bioethics Commission took this on as their first challenge, and they issued a great report on this, that in fact we need a lot of new things for genome design, including self-destruct mechanisms in synthetic cells, limiting where they can grow, et cetera. But also it's becoming an important part of, unfortunately, tracking things for biological warfare. And there's a report you can get from the jcvi.org website that Bob Friedman led. It's a Sloan Institute-funded study, involved my institute and MIT, looking at the security issues. And basically, the federal officials involved in different security aspects said right now there's so many different ways for somebody to get something harmful biologically. You can go to any farm and get anthrax if there's a dead cow there. You can go to probably 1,000 different freezers and find very key lethal viruses. And it doesn't mean it's not going to be a problem for the future, and it has to be built into the software control. Means your antiviral software has to be really good if you're trying to download a vaccine and somebody sends you something else. So it's going to be an important part of how the world is changing.
Thank you. I just had a question. You said you hope to sort of arrange the genome in a functional way that's not the messy and unsorted way that it sort of naturally occurs. And I'm wondering, how do you go about that when some genes have perhaps multiple functions that are unrelated to each other? If one gene is related to neural development and to something trivial like fingernail growth, how do you go about functionally arranging the genome?
Well, I think it was one of the early surprises to people when we had the first human genome that it wasn't functionally organized, okay? You don't have a chromosome for your brain, you don't have one for your fingernails, you don't have one for your heart. Even multi-subunit receptors. I think it was a big surprise early on in neurobiology when it was found that the subunits of the nicotinic receptor were spread on different chromosomes. So it's not organized. It is kind of random because there's these random recombinations. So we're trying to see if we can make it more efficient and enable design by getting it to a cassette level. But there are going to be empirical experiments, and it could be a long time before we're able to solve all those things. There's one in the front here.
All right, this is going to be our last one.
No pressure, but this is going to be really profound, right?
Not quite. In your presentation, you kind of touched upon bioteleportation and its various uses. I was wondering, do you personally have any plans to contribute or advance this technology?
Everything we're doing is inventing the technology and advancing it. So I'm not planning on teleporting myself, although a lot of people are concerned with that because with my genome being the first one sequenced, it's been broadcast into space now for over a decade. And the biggest concern is there's going to be ships coming with tens of thousands of Craig Venters coming back to take over the planet. So we're trying to advance it every way we can. You'll be able to buy a teleportation machine from us in the near future.
Well, I don't think tens of thousands of Craig Venters would be a terrible thing. But so with that, please join me in thanking him once again. Thank you for coming.
Thank you very much.
That was fun.
Yeah.
Absolutely fun.